Hello, my name is Gabrielle Hetzel and I'll be addressing Saving the Bees, a Pollinator Problem, and how do pesticides affect honeybees and their behavior. So why are bees so important? I'm going to start off with a quote from Albert Einstein. He said, "If bees disappeared off the surface of the globe, then man would have only 4 years of life left. No more bees, no more pollination, no more animals, and no more man." Now we have an understanding on how vital honeybees are to our ecosystem. It is important to see how pesticide exposure could be linked to the decline in global honeybee population. We lack the knowledge to see how much pesticides affect bees and their behavior. Immediate action must take place to prevent the devastating effect of pesticides on bee population. So Save the Bees is not only a catchy slogan, and this is why I'm presenting my research question, how do pesticides affect honeybees and their population? And that brings us to our experimental research design. Because honeybees have an important economic role of producing honey, beekeepers thrive off the commercial use of honey and other beeswax programs. Luckily, there is an apiary registration which keeps a database of registered beekeepers in each state of America. Ideally, we would like to get into network and have 10 beekeepers randomly selected from different states of the United States. This is important because it will provide diversity in location and climate, which includes weather and humidity. So the bees will be under different climate conditions throughout the duration of the study. Each beekeeper will provide 3 sets of different colonies, approximately 1,000 bees that will be kept in separate wooden cages. The colony composition would be 1 queen bee, worker bees, and drones, which is the natural composition of colonies. So in result, we're asking for 10,000 honeybees in total from different locations around the United States. Because we are investigating the effect of pesticides, we have chosen to go with the pesticide that's most commonly used not only in the United States, but globally, which is the neonicotinoid. We will also be controlling the pesticide exposure. The pesticides will be used throughout the experiment to see the side effects of pesticides on the entire honeybee colony. And the exposure will consist of 2 different ways. The first way is the neonicotinoid pesticides that will be introduced to the honeybees as an actual direct contact exposure. So this means that they're physically being sprayed with the pesticide, and that they will be in different durations and different variables. One colony will also be exposed to the neonicotinoid in oral exposure, which means that the colony that is exposed through oral exposure will be provided their normal diet, but will have a combination of the neonicotinoid in their diet. And then the third colony will be our control group, which means they will not be exposed to any neonicotinoid, and they will be the control group from the other 2 colonies. I have provided this slide with our experimental design that provides a table that is going to show the different variables. So there will be a total of 3 different variables in the experiment. The goal is to determine the effect on these. So we will be routinely monitoring 3 different colonies from 10 randomly selected beekeepers. We also will manipulate which colonies undergo exposure to the neonicotinoid pesticide, and whether it be direct contact, oral exposure, or our control group. And as you can see, we have this table that is setting a schedule, which is establishing these 3 different variables between beekeepers. So you can see beekeeper 1 and 2 will be daily exposure, 3 and 4 will be weekly exposure, 5 and 6 would be biweekly, 7 and 8 will be monthly, and 9 and 10 will be yearly. This is important to establish because then we have a consistency and a schedule that we will be following. Most of the honeybee behavior monitoring will be visual analysis of each colony. In this experiment, there will be multiple colony level exposures and continuous monitoring. The control colonies will give a baseline on honeybee behavior and the expected honey production. Other factors that we will be measuring is the colony death rates, illness, as well as honey production. So the colony population will be tracked over the entire duration of the experiment, which will be the duration of 2 years. So in those 2 years, the honey production will be measured monthly, and this will be done by weight. Also, another measurement that we will be doing monthly is the refractometer, which measures the moisture level in honey. So in a healthy colony, honey is moist, and in ill or not well colony, it's usually dry, and the way we'll be measuring that is with the refractometer. Any illness or abnormal behavior will be collected in the data analysis weekly, and the data analysis will determine if pesticide exposure to honeybees will have a negative effect on the colony. And this data analysis in the experiment is vital to see the short-term and long-term consequences of pesticide exposure to the colony development, behavior, and survival. Thank you so much.